Thank you very much, Dr. Mao, for the introduction and the opportunity to participate in this really inspiring session. So I'm going to start with my motivation of how we go across our problem. So in my case, I'm primarily interested in biotechnology as treatments, not pharmaceuticals. And so for us, our digital twin modeling is mainly focused upon a full end-to-end engineering approach, one in which we start with a mathematical model of the system that we're going to fit to our data, the ability to now do optimization on that model, and then finally the ability to turn that optimal solution into a biotechnology framework. Specifically, I'm interested in neurostimulation. And so in this case, our treatments are not valued as milligrams of dosing some pharmaceutical cocktail. Our treatments are valued as either a waveform that goes into an electrical stimulation device, or for the most advanced technology now, we're talking about things like feedback policies, where we have an interface between the device, it's going to be reading some signals off the body, and then it has a rule internally that's going to determine how it sends an electrical stimulation to the organs. And so our focus really is in terms of dynamics, like the wonderful talk by Dr. Yang. And these, of course, occur across different organ systems, not just the brain. So all of our electrical organ systems, the skeletal muscles, the heart, all of these are going to be fundamentally shaped by the dynamics of that organ, and that's generated intrinsically, right? This is a property of the organ. It's not just the time-to-time variation over days. And that's going to shape how an electrical stimulation or any other external perturbation is going to interact with it. And this really goes end to end, right? So we can start with a disease, let's say Parkinson's disease for the brain or bradycardia for the heart, right? That disease is fundamentally shaped by changes in the dynamics of the organ. That's what is the physiological mechanism that's ultimately affecting function, not just the chemical changes or the genetic changes that gave rise to it, but that's really where the core of the impairment starts to happen. And then once we get to the level of device, right, that device is characterized by the dynamics, by how we interact with it. So this is in the framework of I'm going to change the stimulation frequency. Maybe it's a pure sine wave, maybe it is a coupled waves of different amplitudes, or maybe it's even a more advanced device that we have now where we have closed loop feedback. In all these cases, what we're talking about is changes in the dynamics being both the disease pathology and the objective of how we're going to try to change it. Right, so now that we know what we're trying to do, I'm going to start thinking about what are the types of digital twins that we can use to treat it. So we have different levels of biological realism we might start with. We could start with a fully unconstrained approach, either using MLPs or the newer CAN frameworks, going all the way to, on the other opposite spectrum, a purely biophysical approach in which we have all the molecular details put in. Because we're working really on single-person data, the approach that we're going to take is a constrained approach. So this is one in which we work with a model that is an abstraction from the biology, right? So these will be things that try to integrate the most fundamental mechanisms, so things like the population-level activity of neurons as opposed to the individual chemical mechanisms within a neuron. And the fundamental reason why we go with a biologically informed as opposed to a pure end-to-end machine learning approach is the issue of being able to express different interventions within our model. Right? So if we go to an end-to-end machine learning approach in which then we're fundamentally limited to the feature space of the data. So if I am taking electrical recordings from a patient, right? Maybe I have like 64 channels. Then I want to try to make some inferences. Okay, what happens if I apply a, let's say, transcranial magnetic stimulation? This patient's also maybe taking some other pharmaceuticals, maybe a psychotropic compound like Seroquel, right? All those other things can't necessarily go into that model without having to add on additional things to figure out where it goes. As long as we're within a biologically informed format, one in which we have meaningful units, we can still use, basic relationships in physics to try to incorporate different treatments to be able to identify optimal interventions without having to go quite to the level of the exact mechanisms going on at single cells. Another important feature is that when we're talking about neurostimulation or even if we're using feedback devices, we have a very different space of interventions than if we were just talking about optimal dosing pharmaceuticals. Right? In this case, our intervention space is a space of waveforms or a space of feedback policies. So this is a space that's infinite dimensional. We don't really even have data to train models on for this sort of space. No one's like randomly zapping people with randomly shaped waveforms and then recording the data for public use for us to train an end-to-end approach on. So this is really, I think, the best approach for the cases in which we're trying to optimize functions as our intervention, as opposed to saying optimizing a discrete valuing of, say, pharmaceutical dosing. All right, so we're going to use a biologically informed framework, and so then we have to decide, okay, we're not quite at the level of all the individual mechanisms within cells, but we still want to have the essential features that will allow us to replicate the mechanisms underlying brain dynamics. And so this is going to involve the interplay between two levels of organization. One is the local networks of brain cells. So this is where we start talking about types of neurons connecting, or neural populations, as well as the gross brain anatomy of how different areas of the brain connect to each other, which we call the connectome. And they influence each other in the following way. So brain oscillations fundamentally start as the interaction between two types of neurons, excitatory and inhibitory neurons. They communicate via glutamate and GABA respectively. The excitatory ones increase neural activity, and the inhibitory ones decrease neural activity. And so the back and forth feedback between these is what generates an oscillation that we observe in the brain. And then the connections at a much larger spatial scale that we can observe using diffusion imaging between brain areas, these are what allow these oscillations to pass over the brain, to reverberate and form things like traveling waves. Now, they're not separable. So the way that a traveling wave will propagate will depend upon all the local properties of the areas that it's hitting, right? So as it passes through one area, it'll filter in different ways. So the relative strength of excitatory versus inhibitory coupling in a brain area, that'll give it resonance. And so that'll affect which sorts of frequencies it's sensitive to. Now, the problem when we want to try to get to doing these biologically based models is that we don't actually have the ability to directly interface and measure with them. So we've got those two populations, excitatory and inhibitory, right? So this is a fairly high level representation of neural architecture. It's very simple, and yet it's still actually not the level that we can measure. And the reason is that the way we measure brain waves is due to a very convenient fact that the excitatory cells are shaped like triangles, right? So they sort of have like a top up here, and they have a soma at the bottom, and that generates a charge differential that we can measure. So excitatory cells are the only type that generate electrical signals that we measure. The inhibitory cells, the other half of our oscillator, is totally silent when we measure it from a distance non-invasively because the cells are symmetric. And so the problem is that when we try to do this type of estimation, we only can measure signals from half of the system that we want. This is in the best case scenario. So we're totally ignoring things like spatial resolution, signal propagation. We're only talking about fundamentally what is the hard limit that at the very best case I'd be able to do. In this case, it's still ill-posed if we just look at a single snapshot. So I want to make this concrete in terms of what data we have available, and then I'll talk about how we solve this. So we start with a digital twin setup having two types of model. The first is a measurement model. So this tells us where our data came from. This measurement model we build from individual subjects' MRIs. We're working on ways to try to use this based upon template MRIs and personalize it to try to reduce costs and increase applicability. But fundamentally, we can take an MRI, we can segment the different parts of the head, different tissues, scalp, cerebrospinal fluid, brain, skull, right? These all have different conductivities, and we can build an electrical model, and this problem is basically solved, that will tell us how an electrical signal on the brain would propagate to the scalp. So this is our forward model. We usually measure this using electroencephalography, so EEG. In the clinic, this might be 16 to 32 channels. For research, it's usually more common to have 32 to 64. The higher end systems now are around 256. Right, so this is a forward model. This is solved. This is how our data was generated. And then we have the state space model. And what I mean by state here is a spatial pattern of activity over the whole brain. So this is a continuous valued state, right? Not the quantum discrete states that we'll hear about this afternoon. Now, the state space model essentially takes the form of what we would call now a vanilla recurrent neural network, right? These are population level activities of the brain, but they have the same mathematical form as, say, a RNN model for machine learning. with some constraints that now we have the fact that only certain cell types can connect to other cell types, things like that. Right, so you've got a combination of, on the most left-hand term, we have the wiring between the different brain regions, and that is specific to different cell type targets. And then the middle terms, we have the local circuits between the different cell types. These are what's fundamental to generating those oscillations as opposed to propagating them across the brain. And the rightmost terms are just time constants. These describe how quickly activity is integrated within each brain area. All right, so that's the problem setup. We've got a system in which we can, at the very best, measure half the variables. And so the question then is, how do we fit a model like this? How do we measure, how do we invert both the parameters of this system to make a digital twin model of an individual, as well as to track the different types of brain activity once we have a model built? So I'm going to start with half of this problem, right? Because there are two halves. So if we were in the case that we already knew the model and we just wanted to try to solve the activity, we actually have an approach to do that already. It's called the Kalman filter. So what the Kalman filter does is if we have a model of the system, a good model, and we have a time series of measurements, then we can infer what those latent states are. And it works like this. We start with, on each time step, a distribution of our prior expectation of the different states they could be in, right? So the likelihood of it being in a different pattern of spatial activity. We take a model. The model lets us make a prediction. So the model predicts what's the distribution at the next time step, right? So evolving a distribution, what's the brain in at time step k? Okay, now what's it in at time step k plus 1? And using the models from the anatomy, we could then make a distribution of measurements. What the Kalman filter does is it allows us to take those measurements that we just acquired, and these measurements, again, are going to be in the space of scalp EEG. What it allows us to do then is to take those and invert those back to figure out what the true brain activity would have been. So there's a lot of math when the Kalman filter is usually presented, but it actually just turns out to be linear regression. The difference being that when we make this regression model, the Kalman filter is based upon known distributions, right? So I know the distribution, I can exactly calculate what that linear regression matrix should be, and then I can apply it to this, right? So I never actually have access to the distribution of the brain activity itself. I build a model that tells me that distribution. The Kalman filter takes that. Now I can infer what the brain activity was that I indirectly measured. Now that's if I had a model. The problem is that we don't have a model to start with, right? So we have half the problem would be solved using classical, engineering estimation if we did know the model. The question then is, can we use methods from modern machine learning then to train a Kalman filter, right? And that's what we're going to propose, is training a Kalman filter, so that way we're simultaneously solving for the digital twin model parameters, all the brain connectivities, as well as the activity of the different cell types that we couldn't directly measure for both types in vivo. And so if we look at a Kalman filter algorithm, it's a recursive algorithm. So every recursive algorithm we can sort of think of in the same sense as a recurrent neural network. So what we do is we set up this sort of architecture in which we turn the Kalman filter stages into layers of a recurrent neural network. Now these are not standard layers. The initialization to build a prior distribution, that's basically just simulating an RNN with noise. That's normal. Things like the Kalman filter update, these involve operations that you won't see in a normal RNN, things like the inverse Cholesky decomposition. But in the grand scheme of things, we use neural network sorts of algorithms. We do things like gradient clipping, all of our normal backprop routines, things like that. And we can train models now that were operating in spaces that we couldn't directly measure all the variables. Now, the key difference here, though, is that while this takes the form of a recurrent neural network for training, this is not a black box. There are no uninterpretable parameters here. Every single layer of here is totally parameterized by a biological model. Everything from what are the dynamics within the layer to how does that layer map onto measurements. All of those are totally determined by the biology and by the characterizations of what we're trying to model. So let me show you just at a glance what the different things that come out of there are. So we get things that are both connectivity maps between the different types of neurons in the brain. So here I have two maps for each person, an excitatory map and an inhibitory map. So these are all the connections between brain areas. And we also have spatial patterns of activity that describe the different local circuit parameters. And we have simulated dynamics here just to give you a sense of what they look like. One thing I'll point out is that neurologists often indicate that this is actually the most convincing piece, not the quantitative stuff, is that this actually looks like a real EEG from a human subject. We see the most prominent features, alpha waves, beta bursts, they're localized where they should be. So these are actually quite accurate models. We compare these with some existing methods for this sort of problem. Now, these methods do not scale, but even within the range of when these methods are applicable, things like expectation maximization, joint and dual filtering, our method well outperforms them. It scales with a much lower complexity. So talking about like 15 minutes versus 250 hours. And it's more accurate in general. So here's some validation. This is with the Human Connectome Project data. So we want to see, are these reliable? If I take a person in and I scan them twice, do I get much more similar models for them than for another person? So on the left-hand side here, this is what we call a fingerprinting plot. It's just showing along the diagonal here that models fit within a subject are much more similar than between subjects. We see that the spatial localization of different EEG power bands are consistent with what we know from data, and that we correctly predict the individual differences between people in the different EEG spectral power. So this is showing you how strong different frequencies of oscillations are between individuals. So all of that's well predicted. We're well characterizing the different spatial and temporal characteristics of every person that we fitted. So it's mainly focused on methods. So we're doing some things now looking at genetics. So we're showing here on the left-hand side that when we're looking at twin studies, we can find that the parameters are quite heritable. So we have roughly double the heritability for monozygotic versus dizygotic twins. We're doing some applications to the ICU. The top here should be a GIF, but that's okay. We can do things like we track a patient over a multi-day ICU stay. We can see where there are changes in the digital twin model estimates. So those are indicating where there was a change. Here, in this case, this person had cardiac arrest. There's a sudden change. And then there's also a period in between where there's a more subtle change later after the cardiac arrest, in this case, as well as some applications now to identifying person-specific optimal TMS strategies. So the future things we're looking at is actually implementing this in hardware. So we've developed some new hardware to actually do closed-loop stimulation. So trying to do this online, build models, stimulate, use those to inform models, and ultimately build closed-loop systems for personalized stimulation, as well as developing resources for the community. So we have some collaborators that are actually building mixed reality models of the brain for epilepsy patients. And so what we're hoping to do is include these in pre-surgical planning. So right now the system allows them to see the existing data in 3D and interact with it. What we're hoping to do is to now be able to simulate interventions. So practitioners can see, what would happen if I did this? I can see it on the brain of the person. If I am involved in regulatory sciences, I can actually simulate testing out a device on a person without actually having a patient in front of me, as well as the more basic mathematics of how do we now go from describing these very fast brain oscillations to thinking about changes over much slower time scales. I'd like to thank some of the previous funding sources that supported this: National Institute of Health, National Science Foundation, data from the Human Connectome Project. We also have support from the James McDonald Foundation and the Beckman Institute.
So, thank you, Dr. Singh, for wonderful presentation. So I believe every people body is getting old. A brain model, a brain digital twin will be helpful for like Parkinson's disease, Alzheimer's disease. Everybody has probability to get disease like that. So we have time for one question.
Huge need. So the goals there, I can't speak for the method, but it sounds logical. Limited understanding, so you'll have to excuse me. But when it comes to once you have a working model and then you're trying to use it in contexts such as interventions, how are you thinking about validation to the extent that you can validate something like this?
Yes. So the nice thing about these sorts of dynamical systems models is that the predictions are extremely specific. The prediction is of the form, if I pull somebody with, let's say, a theta burst TMS pattern, this is the EEG signal that you will measure at every single channel of your EEG. So this is engineering, type levels of prediction. This isn't, I'm going to correlate that alpha power will go up or down. I'm saying this is exactly what I believe will happen. And so for the TMS things, we've had two types of validation. So right now, the validation has been at the parameter level. So we've shown that we can track the course of changes in relative excitatory and inhibitory activity for different types of TMS. So there's one type that's thought to promote inhibitory changes in brain plasticity, another type excitatory. We've shown that what we see is that if we fit the models at different intervals afterwards, we see like a gradual change as the brain changes post-treatment. What we're going towards is actually the case in which we're making those specific predictions. This is how the brain's raw EEG values, so the electrical activity, this is the time course that it will take. And we're hoping to use engineering optimization methods then to try to actually identify, okay, here is the optimal waveform to input if I want to do something like, say, increase this type of oscillation. So the validation can be at the level of parameters, which is sort of just based on prior scientific assumptions, or at the level of time series, in which case you're putting all your dice on the board and saying, this is exactly what you will see.
And those are not altered then in disease states. So you would, so for example, in a disease state, and you're applying the same, we'll say, I'm going to reduce it for my own understanding.
Sure.
Electrical stimulation, you know, I'm going to apply this and I predict based on this model that I have that it's going to take this path, et cetera.
Yes.
And that is consistent across disease states. Is that what the assumption is?
So there isn't an assumption of a disease state. So this is supposed to be general. It's not based upon a model of a specific disease state. This is for a person in isolation. And so it's not premised upon us knowing what it is that's wrong with them. It's premised upon us saying, this is what I want to change about this person.
Got it. Thank you so much.
Yeah.
Okay, before we conclude, I want to turn over back to Nick. He may have some logistic item.
Thank you, Feng Lao. Thanks for all the speakers. One more time, give a quick hand.